Hello grade 12, so tomorrow is your life science paper 2 finals paper, and this is just a video on genetics and inheritance for revision. Here's the department's guidelines, but as usual, everything in the guidelines has been mentioned in the PowerPoints. So an introduction. Gregor Mendel is the father of genetics, and he conducted experiments with peas, and from these experiments he created two laws, the law of dominance and the law of segregation. Then we get to concepts in inheritance. We have terminology, so a few words that we need to know before we start the chapter, but they will become more clear when we go through the work, but this is just revision, so you should know this already. Heredity refers to the transmission of characteristics from parents to their offspring. Inheritance refers to the set of characteristics that have been passed from parents to offspring. Genetics refers to the study of heredity and the variations that occur in the transmission of hereditary characteristics. Genotype refers to the genetic makeup of an individual. The phenotype is the external appearance of an organism, and this is determined by the genotype. So the genotype influences the phenotype. Alleles, they are alternate forms of a gene that control the same characteristic, but in different ways. Dominance describes a gene whose characteristic always shows up in the individual, either in the homozygous or heterozygous combination. And it does this because in the heterozygous combination, it masks the recessive allele. So in the phenotype for homozygous and heterozygous combinations, the dominant allele will always appear. That's for the phenotype. For recessive, it describes genes whose characteristics only show up in homozygous combination, as it is hidden or masked by the dominant allele. Heterozygous, an individual which has unlike genes for particular characteristics, so this will be like big T, small t, and homozygous, an individual which has similar genes for particular characteristics, so big T, or small t, small t. Just an example for like if we're doing height and big T is tallness. Okay, then Mendel's law of dominance. When two homozygous organisms with contrasting characteristics are crossed, all the individuals of the F1 generation will display the dominance trait. What this means, I showed a Punnett square here to basically describe what this is saying. So two homozygous organisms, capital B, small b, with contrasting characteristics are crossed. So if they have contrasting characteristics, this one has the capital B and the other one has the small b, the lowercase letter b. And all the individuals of the F1 generation will display the dominant trait. Why? Because all of these individuals are now heterozygous, and there's the dominant allele which is masking the recessive allele. So in the phenotype, the dominance trait will be shown. The big B will be shown. An individual that is heterozygous for a particular characteristic will have the dominance traits as the phenotype, which we just said now. Okay. Then Mendel's principle of segregation. An organism possesses two factors, which separate or segregate, so that each gamete contains only one of these factors. What Mendel is describing here is basically just meiosis. So when two gametes are formed, or when the gametes are formed from meiosis, one will contain one of the factors and the other gamete will contain the other factor. So if we're dealing with the B's in the previous slide, big B, small b, the one gamete there'll be a big B, and in the other one there'll also be a capital B, and then in the other parents there'll be a small b and a lowercase letter b in the other gamete. Okay, so then monohybrid cross. Monohybrid cross it shows the reproduction between two individuals where the concern is with inheritance of only one characteristic. And then we did Mendel's principle of segregation in the previous slides. I'm just going to go here onto the table. The table shows three kinds of dominance that we get: complete dominance, incomplete dominance, and codominance. For complete dominance, we have, for the alleles, one dominant and one recessive allele. And in the heterozygous condition, the dominant phenotype is shown. So if we look here, to represent complete dominance, we use a capital letter and a lowercase letter. So for the genotype here, for red is now the dominant trait. So a capital R and a capital R show red, a lowercase letter R and a lowercase letter R show white. Notice that we don't use two different letters here. We use the same letter in as a capital letter and as a lowercase letter. And then when we have the heterozygous condition, a big R and a small r, we get red. So we have red plus white to give us red, because this is heterozygous and the dominant allele masks the recessive allele. So it will show up as red because red is the dominant trait. Then we get to incomplete dominance. So the word kind of gives us an idea of what it's saying. Incomplete dominance means that nothing is dominant and nothing is recessive. And then in the heterozygous condition, a third new phenotype is shown. The keys that we use to represent it, so we use two letters now. For complete dominance, we only use one letter. For incomplete dominance, we use two letters, and for codominance, we use two letters. So yeah, RR shows that it's red, and WW shows that it's white, and then in the heterozygous condition, RW, it makes a third new phenotype, which is a mixture of the two. It makes pink. So yeah, you can see red and white makes the pink. So like basically colors. Green is made by blue and yellow, you know, those kinds of things. So it's a mixture of the two. In codominance, both are equally dominant. So in the heterozygous condition, both influence the phenotype. Two letters are chosen as well. RR shows that it's red, WW shows that it's white, and then RW now shows red and white. So both show up. It is now a combination of the two, not a mixture. It's a combination, red and white, because both are equally dominant, so they both show equally in the phenotype. And then this is the format of a genetic cross, and we get a few examples here of the genetic cross. So for complete dominance, we have two hybrid tall trees. What does hybrid mean? It's another way of asking you heterozygous. So two hybrid tall trees are crossed with each other, and if they are heterozygous and they are tall, it means that tall must be the dominant allele or the dominant trait. Okay, and then use the genetic cross to show the possible genotypes and phenotypes of the offspring. Also indicates the genotypic and phenotypic ratios. Okay, so we have the genotype. Like I explained just now, it's heterozygous, and because they are tall, it means that the tall is the dominant trait. So big T, small t is what I use to represent that, and then big T, small t, because they are two, and they are crossed. We have the phenotype tall, and this is also tall, and then meiosis takes place, and the gametes have a big T and a small t, and a big T and a small t. Then when we get to fertilization, here are our gametes, the big T and the small t, for the one parent, and the big T and the small t for the other parent. Big T cross together to make big T, big T. Small t, big T cross together to make big T, small t. And you always put the capital letter first, okay? Big T, small t, heterozygous, small t, homozygous for the recessive condition. Then by our genotype, we have one of these, two of the heterozygous, and one of the homozygous recessive. So the phenotype for these, this is tall because the dominant masks the recessive, and this is short because both are recessive. And then the genotypic ratio, for this it's 25%, because our ratio is 1 to 2 to 1. And if we say 1 over 4, because we have 4 possibilities, 1 over 4 times 100 is 25%, 2 over 4 times 100 is 50%, and then 1 over 4 times 100 is 25%. And for I didn't put it here, but the phenotypic ratios, it's 75% tall and 25% short, because 3 of these are tall and this one is short. So 3 over 4 times 100 will be 75%, and 1 over 4 times 100 will be 25%. So the phenotypic ratios, 75% can be tall and 25% short. That's the chances of it being tall and short. Okay, then we get an incomplete dominance example. In a certain species of flowers, there's incomplete dominance between the alleles for red and white flower color. This leads to the production of a third phenotype of pink flowers as an immediate characteristic. Represents a genetic cross for the crossing of the red and white flowering plants. So here we have RR and WW, two different letters in this case because it's incomplete dominance. In the phenotype, we have red flowering, and this one is white flowering. Gametes, RR, and the other ones will be WW. Then fertilization takes place, so our gametes, RR and WW, and when they cross, all of the offspring are now heterozygous. So in the genotype, all of them will be RW, and in the phenotype, they will all be pink because it's incomplete dominance, so it will be a mixture of these two. Red and white will make pink. And the ratio will be 100% because all of these offspring will be RW and all of them will have a pink phenotype. Okay, so then codominance. A farmer mated a black sheep with a white sheep and obtained, there should be lambs with a mixture of black and white coat color. Represents a genetic cross for the above results that were obtained. So big W, and big B. Again, two different letters because it's codominance. The phenotype, white and black. So we have gametes, WW, and B. And then we have our gametes here again during fertilization in our Punnett square. And when they are crossed, they make WB for all of them. So then the genotype of the F1 generation, which is the offspring, the P1 generation are the parents. So in the offspring, all of these will be WB, and for the phenotype, they will be black and white. Why? Codominance, so the white and the black are equally dominant. So in the phenotype, it will show up as black and white. It will not be a mixture of the two like in incomplete dominance. It will not make gray. It will be a full combination because both are equally dominant. So black and white. And then 100% of the offspring will show this, because here all of the offspring show WB. Okay, then we get to sex determination. So in humans, there are 20 pairs of chromosomes, and these are autosomes, and then there's one pair of chromosomes, which are sex chromosomes or gonosomes. Males have XY chromosomes and females have XX chromosomes as their gonosomes. And then there will always be a 50-50 chance of the offspring being female or male. Because in males, the X and the Y chromosomes, so sperm either have an X or Y chromosome, and the sperm with X chromosomes are equal in amounts to the sperm with Y chromosomes. So there's always a 50-50 chance because women they only have X chromosomes. So if fertilization takes place, and yeah, you can see it in this Punnett square, there's an X and an X and an X and a Y. So the X and the X makes XX, this X and this X makes XX, this X and this Y makes XY, and this X and this Y makes XY. Yeah, you can see 50% for girls and 50% for boys. So there's always a 50-50 chance of offspring being male or female. If they ask you, or if they tell you that there's a family, the parents had 3 girls and 1 boy, and they tell you that they're having another child, what is the chance that the child is female? Always 50%. It's not more now because they've had more girls than boys. It will always be 50%. Okay, then sex-linked characteristics. So we look at how sex-linked disorders occur. And the 2 sex-linked disorders that we deal with in this syllabus is colorblindness and hemophilia. So males only have 1 X chromosome. And if this chromosome carries the recessive allele, this now is an example for colorblindness, so the B shows blindness, the male will have the disorder in the phenotype. And this is because the Y chromosome in males does not carry an allele to mask the effects of the recessive allele. So if a man has the disorder on his X chromosome, there's nothing to mask this disorder because in women, females have 2 X chromosomes. They need to have 2 recessive alleles to be affected. A dominant allele on the X chromosome will mask the effects of the recessive trait. So women have 2 X chromosomes. So if there's a recessive allele and this one is dominant, like here, this woman will not show the disorder because she has a normal dominance trait masking this disorder. So it will not show up in her phenotype. She will be a carrier for this, but it will not show up in her phenotype. But if a man has it, because he only has 1 X chromosome, and if this X chromosome has the disorder, the Y chromosome cannot mask anything because the Y chromosome is much smaller than the X chromosome. And the Y chromosome, if you think about a locus is a position on a chromosome where there's a gene. Because they are different in size, the X and Y chromosomes, if there's a locus with the recessive trait for this disorder on that X chromosome, there's no locus on the Y chromosome that is able to mask that X chromosome's locus, that recessive trait. Okay, so if we look at this Punnett square, then we can get a better understanding. This is now for hemophilia. There's the 2 XX, so this is the woman because it's the 2 X cells, and then there's this 2 sperm cells, an X and a Y. The woman is normal on the 1 gamete, and then she has, she's affected here. So she's a carrier for this because she does not show it in her phenotype, but she does have the recessive trait on one of the X chromosomes. The man is normal, he does not carry the disorder at all. So when these 2 X chromosomes combine, the female that is formed, XX female, will be normal because she has 2 normal X chromosomes. This one will be a carrier but will not be affected. She will not show it in her phenotype because she has the 1 recessive trait, but this X with the dominant trait is masking it. So she will not show it in her phenotype, but she will carry it like her mother. This one, he will be a boy, XY, and he will not be affected because he will get this X, which is fine. It has the dominant trait. But this boy will be the affected one because he will have this recessive one, and the Y will not be able to mask this. So he will then show the disorder in his phenotype. Then we get to blood grouping. So some genes have multiple alleles and they are found on the same locus. We discussed the locus in the previous slide. And then blood grouping in humans have 3 alleles for the single gene. So blood grouping is like a gene, and then there's 3 things, A, B, and then the I. So there are 3 alleles. Okay, so then we get to the genotypes and the phenotypes of blood groups. The genotype IA will give you A, but then the recessive one is the small I. And the big IA will mask the small I, so it will also give us A. IB will give us B, and in the same case, the big I with the B will mask the small I to still give us B. Then this is codominance, because there's an IA and an IB, and they both show up in the phenotype as AB, so this is codominance. And this is complete dominance. And then this is also complete dominance because we see the recessive small I, and that gives us O. So for one gene, which is the blood group, we have 3 different alleles in different combinations, which give us these. Okay. Then we get to dihybrid crosses. So Mendel's principle of independent assortment says that the various factors controlling the different characteristics are separate entities, not influencing each other in any way and sorting themselves out independently during gamete formation. So basically, with his experiment with the peas, he had yellow and green peas, and one was shriveled and one was smooth or something like that. And he basically says that the color and the shriveledness does not influence each other at all, because you get yellow ones that are shriveled and you get or wrinkled, there we go, wrinkled and round. So you get yellow ones that are wrinkled and you get green ones that are wrinkled, and you get yellow ones that are round and green ones that are round. So they basically don't influence each other in any way. During gamete formation, they split independent of each other. And now for dihybrid crosses, I use the FOIL method from maths to get the different combinations you can get. So you're going to have now 4 columns and 4 rows, because there are 2 traits that you are now dealing with. You are dealing with seed shape and seed color. So here is the round and the wrinkled and the yellow and the green. So you first do the firsts, R and Y, these two, the big R and the big Y, and you get RY. Then you do the outers, the R and the small y, so you get RY there. Then you do the inners, so the small r and the big Y, and you get that. And then the lasts, the R and the Y, to get that one there. And then you do the same for the other one that you have here, and then you cross them. And then it's just very easy. You just put the R and the R together and the Y and the Y together to get your dihybrid cross. And then at the end, you count how many you have of each kind of thing. Here they counted 9 out of 16, round and yellow. They counted 3 out of 16, round and green. They counted 3 out of 16, wrinkled and yellow, and 1 wrinkled green. And then you can work out the ratio by saying 9 divided by 16 times 100, 3 divided by 16 times 100. And you get your ratios. Okay, then we get genetic lineages and pedigree diagrams. So there are 4 things that we need to look at to help us figure out what is happening in this genetic or the pedigree diagram. So we first look at generations. Here's the first generation, here's the second generation, here's the third generation, here's the fourth generation. Then we look at males and females, and we use the shapes in the key. So this tells us that males are squares and females are circles. So now we know this is a male and this is a female, and they, if you quickly look at relations, the vertical lines show offspring, that the horizontal lines show reproduction. So this shows that these 2 people reproduce to give this offspring. And then these 2 people reproduce to give this offspring. And these 2 people reproduce to give these offspring. Okay. And then shading, affected versus unaffected. Here they use different colors. So here for unaffected they use blue, and for affected they use red. So this is an affected female who mates with an unaffected male. And their offspring then give us this. And you can go work out the Punnett squares to figure out what exactly is happening in each of these individuals. Okay. So that's how you read the genetic lineages or the pedigree diagrams. This is a pedigree diagram. Then we get to mutations. So a mutation is a sudden random change in the genetic code of an organism which can be inherited. And mutations introduce genetic variation. So we get gene mutations and chromosomal mutations. A gene mutation is a gene mutation refers to a change in the structure of a gene as a result of a change in the DNA sequence. There are 2 types of gene mutations. Number 1, a point mutation, and number 2, a frame shift mutation. So let's look at a point mutation. A change in a single base pair in the DNA molecule at one point occurs. This will cause a corresponding change to the one base in the mRNA molecule, so the messenger RNA. And the codon in the base changes, the change occurs in the messenger RNA, and this will now be different, which means it will code for a new amino acid. So if you look here, here's the original sequence, and this is now, let's see, this is DNA because here's the T. So we know it's DNA. And there's a change here, the C, where this T was. So there's now cytosine where there was thymine. And this is now going to code for a different mRNA molecule, because an A will form here, U, U, G, and where there should have been an A, there will now be a G in the mRNA, which means the codons will be different, which means new or different amino acids will be formed at that locus. At that codon. So the codon in which the base change occurs in the mRNA will now be different, and this will code for a new amino acid. A different protein will form containing one new amino acid. The new protein may function differently, leading to altered characteristics. Then we get to frameshift mutations. A single base pair may be inserted or deleted from the DNA molecule. From the point of insertion or deletion onwards, the DNA will reproduce mRNA with codons that are now different, since the codons will now read in new frames of 3 bases. So a point mutation is just a different nucleic acid being put in there. A frameshift mutation is one is completely lost, it's taken out. So yeah, you can see CAT, TCA, but now there is one inserted, it seems. So it moves up completely. Now it reads ATC, CAC, and this is going to code for completely new amino acids, which will make a completely new protein. So a new protein will form with a number of new amino acids, or amino acids in a completely new sequence, which is happening here. You can see they're completely different to these ones. Then we get chromosomal mutations, which refer to changes in the normal structure or number of chromosomes. It occurs from incorrect crossing over in prophase 1 or incorrect separation in anaphase 1, which results in non-disjunction. If you don't understand this, then you can go watch my meiosis video. So basically what happens here, deletion, a section of chromatid is lost. So a section of a chromatid is lost. Let's see, deletion here. So this is now completely lost. So this section of the chromatid is gone, okay? Duplication is when a section of a chromatid is doubled. Okay, they don't have duplication on this photo, but you can understand that a part of the chromosome is now more. Inversion is when a section of a chromatid breaks off and reattaches incorrectly. So insertion, this broke off and is now attaching here incorrectly. Translocation is when a section of a chromatid breaks off and reattaches to a different chromosome. They also don't have translocation here, but you can understand that a section of a chromatid, so on a chromosome, it breaks off and reattaches to a different chromosome. And now all of these are going to cause new things to be formed, new amino acids. Like, yeah, you can see this changes from that, and this changes from there, and this also changes from this one here. All of these actually change here with the deletion. Okay, then the effects of mutations. Harmful mutations are lethal mutations which causes a mutated organism to die, and characteristics are not passed on. So basically it's nature's way of making sure that this lethal mutation is not transferred or transmitted to an offspring. So it dies before it can mate and produce offspring, so before it can reproduce basically. Harmless mutations, they have no effects on the structural function of the organism which possesses them. Useful mutations are advantageous mutations, mutant genes, which may wipe out all the alleles controlling the same characteristic within the population. Why? Because of natural selection. So if they have an advantageous mutant gene, they will survive longer. If, let's say, a drought occurs and they have this mutant gene that allows the plants to survive during the drought, they will wipe out all of the other ones and then they will reproduce. So only the mutant gene will now be passed on. Okay, genetic disorders. So there are 2 genetic disorders which are discussed in the syllabus, Down syndrome and hemophilia. So it's caused by faulty meiotic divisions, and it's when an extra chromosome on the 21st pair of chromosomes occurs, and this is called trisomy 21. It's caused by non-disjunction. Then symptoms, mental retardation, hearing loss, heart defects, small mouth and nose, decreased muscle tone, and a depressed nasal bridge. And then treatment, there's no cure, but it is treated symptomatically. And screening, which you can do while the mother is still pregnant to test whether the child will have Down syndrome. You can do genetic testing or a karyotype analysis. Then for hemophilia, it is when there's a recessive allele on the X chromosome or recessive gene on the X chromosome. Symptoms, the blood does not clot properly, which can lead to internal bleeding, and the person can bleed to death. So they inject clotting agents to combat this, or immunosuppressant drugs. And then the screening is also genetic screening on the X chromosome. So specifically on the X chromosome because it is a sex-linked disorder. Then genetic engineering. Genetic modification is the removing of a useful gene from one organism and inserting it into another organism to produce beneficial characteristics. The advantages, production of medication and resources are now cheaper. you can do pest control with specific genes inserted into a crop. So they are now more resistant, using specific genes to increase crop yields, which can help with food security, and then selecting genes to increase shelf life of plant products. There are disagreements to this because people say the long-term effects of this is unknown, which can lead to health problems in the future. And the long-term effects on the biodiversity, we also aren't sure of. It could lead to damaging ecosystems and damaging the nature. People say it's morally incorrect because humans are interfering with nature and they are playing God. Initially, it is an expensive process, and many people may not be able, like countries may not be able to afford this. The economy won't survive, and there's better stuff that the money could be used on. That's what people say. Okay, and then we get to cloning. A somatic cell, which is diploid from tissue, is used. It is then placed into the nucleus. I'm so sorry. A somatic diploid cell is used and the nucleus from it is removed. And then this nucleus is placed inside an ovum which does not have a nucleus. So the nucleus from the ovum has also been removed. And then the nucleus from this diploid cell is placed into the ovum, the empty ovum. Now the ovum is diploid because it has a diploid cell placed into it. And ovums usually aren't diploid, they are haploid because they are gametes. So it basically tricks this ovum into thinking that it has been fertilized because it is now diploid. And this stimulates it to divide to form an embryo. Once the embryo is formed, it is placed inside the uterus of another adult, and the embryo develops and a genetically identical baby will be born. And the benefits of cloning is that more food and milk can be produced if cloned from abundantly providing animals. So let's say there's this like great cow and it makes like so much milk and it's really meaty. So they clone this cow because it provides a lot of meat and it provides a lot of milk. So now there's more of these cows which provide a lot of meat and a lot of milk. And this can help with food security. Okay, then stem cell research. It is undifferentiated cells that can be developed into any tissue type. It is used for therapeutic purposes like regenerating spinal tissue for paralysis or pancreatic cells for diabetes. And then sources of stem cells is the umbilical cord, embryos, and bone marrow. That is where stem cells are. And stem cells, when it says undifferentiated, the reason they can be used for things like this is because they, in labs, they then differentiate the cells to make different kinds of tissues because they haven't been differentiated yet, so they don't have a specific function yet. Differentiated cells are cells that have undergone chemical processes to become a certain cell, a cell for your skin or a cell for your stomach lining or something like that. Okay, so these are undifferentiated. They haven't undergone any chemical process yet, so they artificially make them undergo a chemical process so that they can be used to become different kinds of tissue, which can help for healing people. And then paternity testing. So blood tests can only show whether someone may be the father, because many people in the world have the same blood type, or definitely isn't the father. So if we look at this Punnett square, we have different blood groups, okay? If the first question says that the offspring is a child with blood type B, so we know the mom's genotype is IA/IB, then we can definitely say that this is not the dad because there's no offspring here that has IB/IB or IBI, because that's how we get blood type B. So we know that this is definitely not the dad. But if it says child with blood group AB, so the mom has AB and the dad has AA, so there are now two possibilities for IA and IB. So now we know that this combination is possible. This could be the dad. But we don't know for certain that this is the dad because there are many people in the world who have IA or a different kind of genotype, which would also code for AB, for blood group AB. So this could be the dad, but it's not conclusive evidence. This is not conclusive evidence that this is the dad. But for this example, we definitely can say that this is not the dad because there's no offspring here that can be blood group B. Okay. And then genetic links. Mitochondrial DNA is passed down from mother to child. So analysis of these mutations on the mitochondrial DNA were traced to an ancestral female that existed in Africa, and it shows that all humans descended from her. Why we can trace mitochondrial DNA is because during fertilization, only the nucleus of the sperm cell enters the ovum. So no other organelle is entering the ovum. So the mitochondrial DNA is only from the ovum, so it's only from the mother. And that is why we can trace the maternal lineage.